today's video, we are going to look at the structure and the function of two molecules which are found in your cells, and these molecules are known as DNA and RNA. Now, the first little task I'd like you to do is to draw a little mind map and list as many facts as you know about DNA. And these don't need to be facts which are very kind of complicated or sophisticated. Just tell me anything and everything you might sort of know vaguely about what DNA is, maybe where it's found, where maybe what it does, or any other kind of random facts about this molecule you might have heard of. So I'd like you to pause the video at this point, list your facts, and then come back to the video to carry on. So I'm hoping that you were able to name a few facts. As I said, I don't really mind how basic those facts are. So some people usually able to say that DNA is found inside nucleus in a cell, because that's something you would have come across in your cell biology assignment. Some people say that DNA carries genetic information, which is correct. Sometimes people also say that every person's DNA is unique, and we all have a different set of DNA, which is correct to an extent. There is one exception to it, just to remind you, and this exception is identical twins, because they start off by having exactly the same DNA. So these are some of the facts you might have heard about. If you did maybe biology at GCSE, or indeed at A level at some point in your life, you might know more complicated facts like what DNA is made out of, or what the letters DNA stand for, etc. So I'm going to start with a few basic facts about DNA and just explain why your cells need DNA. So what I've done is I've drawn a sort of simplified version of a cell here, and as I mentioned already, DNA is found inside the nucleus of a cell, so that's what the sort of squiggles inside the circle representing cell membrane means. And then another fact that I wanted to remind you about is you also have another important organelle in your cells, and you have lots of them actually, and they're called ribosomes. And in your cell biology assignment, you should have described what ribosomes were, and also be able to identify what they look like on electronic micrographs. So ribosomes are these really, really tiny organelles. On micrographs, they look like a kind of a splattering of black dots throughout the cell. And the job of ribosomes is to produce proteins. So what connection is there between ribosomes and DNA? Well, the fact is, if I take one of the molecules of DNA and stretch it out, I will see that it's a kind of like a spiral, double spiral. And the fact is that what DNA does, that it codes for proteins. So it contains instructions how to make different proteins in your body, which is what that statement says. Now, I usually give people a little analogy, because what I found was that when I say that DNA codes for proteins, people kind of nod along, but we don't necessarily understand fully what that statement means. So my little analogy is about you going to an IKEA store and buying a flat pack piece of furniture. So I hope a lot of you had that kind of experience. So you bring your box with flat pack furniture back home, and when you open the box, what you'll see is that you have a selection of sort of items, and if you put those items together, you end up with a piece of furniture, maybe a wardrobe, let's say. But you also have inside this pack a little cheaply printed booklet which tells you take this part, part A, and then attach it to part B with screws C. So your DNA is a bit like equivalent of lots and lots of these booklets, which tells your ribosome and your cell how to assemble a protein or proteins. So DNA doesn't contain proteins itself. It's a bit like the booklet in itself. It doesn't contain the actual parts of a wardrobe. So all it contains is just information how to do it, how to make it. Another good analogy, and a lot of books use this analogy, is to imagine DNA as a recipe book. So you have each page for a particular recipe, which you can follow and assemble that dish, right? So your protein would be your finished dish, and DNA contains this kind of reams and reams of instructions how to produce different proteins. So for your body, you need round about 100,000 different proteins. So overall, DNA codes for every single protein that your body needs. So within DNA, as I said, if I stretch out one particular molecule, then you have sections which code for those different proteins. So let's say this first section codes for protein 1, then the next section codes for protein 2, and the section after codes for protein 3, and so on and so forth. So my next question, which I typically ask, is can you think of a name given to those sections of DNA which code for different proteins? So instead of saying here in red codes for protein 1, I could have just given this one word, and it's a word which a lot of you would have heard of, and it's not particularly technical term, it's a term we use in everyday language, and I'm curious if you would be able to come up with this word. So again, would be a good idea to pause the video at this point and have a little think about which word I have in mind. And from experience, sometimes people just get completely flummoxed by this question. They really think it's something super complicated. But usually one or two people in each class actually comes up with the right answer. And the answer is very simple. It's just a gene, right? So when we say to each other, oh, my daughter got my genes, she looks a lot like me. Oh, she must have got his genes from his grandfather, his face just like his grandfather's. So when we use this kind of terms, gene, there is actual scientific kind of term which is attached to this word. So gene, as I said previously, means a code for a particular protein, or it could be a code for a part of a protein, also known as polypeptide. This is because sometimes proteins can be very complex and they need to be assembled from more than one kind of block of, more than one sort of bundle of amino acids or polypeptides. So a good example of this would be hemoglobin. So if you think about a picture of hemoglobin, which some of you used in cardiovascular assignment, that you'll know that hemoglobin actually contains four kind of blocks, four sections, which when you put them together, you get one molecule of that protein or hemoglobin. But in some cases, with more simple proteins, one gene will actually code for one complete protein. So that can happen as well. Now, also, another point I wanted to make is, well, why is it that DNA codes specifically for proteins rather than something else? Well, to understand that, you have to think about what kind of jobs proteins do in your body. And I'm again hoping that you should be able to name a few of these jobs. So, for example, you can think of something called structural proteins. So, for example, your muscles contain a lot of proteins which give your muscles strength, structure, and also give your muscles ability to contract and relax, which is obviously quite important for movement, etc. But also proteins do a lot of other jobs. For example, you might have protein channels in your cell membranes which allow substances to go in and out of a cell, and you can control what comes in and what comes out. So that's another really important job. A couple more examples would be enzymes. So all enzymes are proteins, so it's really important for speeding up reactions. And also a lot of the hormones are also proteins. So if you think about all these different jobs which proteins do in your body, so if you didn't have proteins, you wouldn't be able to move, you wouldn't be able to transport substances, you wouldn't be able to communicate between different parts of your body, and you wouldn't be able to speed up reactions. So basically, they're really, really, really important for keeping you alive. And proteins, moreover, need to be very, very particular shape. So think about hormones or enzymes. You should be able to remember that these things they rely on something called lock and key theory. So they need to be very particular shape for enzymes or hormones to work correctly. And the same is true for protein channels or structural proteins. They also need to be very particular shape. And so DNA needs to be able to code for these proteins correctly in order for their shape to be right. So that's why this information is highly important, and it's recorded in this important molecule, DNA. So going back to this second diagram, so I already mentioned that the word used to describe a copy of, the word to describe a section of DNA which codes for a protein is known as gene. And let's imagine that your cell at certain point needs protein number 2. Well, what I can't do is I cannot drag the whole of DNA molecule out of the nucleus and then run it through ribosome to assemble that protein. So ribosome can read that instruction and assemble protein, and then drag that DNA back into the nucleus once I'm done. So this would not be very kind of efficient way of doing it, or very useful way of doing it. So nature came up with an alternative solution where instead of taking the whole of DNA out of the nucleus, and assembling this protein, what you do instead is you just make a copy of this one particular gene which you needed at that time. And then you can use that copy to take it out of the nucleus and then assemble your protein using ribosome. So it's a little bit like going to a big library with lots and lots of recipe books, and instead of taking all of those recipe books home with you and just ultimately, only cooking one dish, you just find the book you need, you go to a photocopier, photocopy this one page, that this one recipe you're interested in, and then just taking it home with you. Yeah, so instead of taking the whole library with you, just take this one recipe. So that's what cell in essence does, makes a copy and then uses this copy to assemble your protein. So this is that first few slides were just to help you kind of understand what DNA is and what it does. By the way, I sometimes do ask my students if they know what the name of this copy is, and the name is actually RNA or mRNA, more specifically. We're going to talk about mRNA in a lot more detail when we get to protein synthesis. So at the moment I'm just mentioning it fairly briefly. So a few sort of key facts about DNA and RNA, which you're welcome to copy in your notes if you would like. So genetic material is found in these two molecules, and DNA and RNA are abbreviations, so therefore three capital letters, but they stand for the full names of the molecules. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid, and we already gave definition of the genes. So a couple of interesting facts about discovery of DNA. That was done by these two gentlemen, James Watson and Francis Crick, in around 1953. They consequently got a Nobel Prize for their discovery. However, one person who got overlooked in that Nobel Prize decision was a female scientist, and you know that in those times female scientists were not always credited with their contribution, unfortunately. But her work was also really crucial for Watson and Crick to be able to come up with a theory of DNA structure, and it's Rosalind Franklin. So if you look at more recent publications, books, she is often also credited with discovery of DNA, as she should be. So I just wanted to point out that structure of DNA is a very complex subject. And so to simplify things, you might find that some of the drawings are more kind of, more look a little bit like this kind of kid's drawing of a house where everything is just a square and a triangle to represent roof and the house. Whereas in reality, behind, if you like, that drawing, the actual truth is a lot more complicated. So just keep it in mind that particularly initially, some of the drawings we're using might be more simple, but as we're going in depth, we will find out that each shape actually represents something a lot more complex. So when you think about the grades, in a sense, I'm happy for, let's say, pass and a merit for you to be able to describe DNA in this more kind of simplified way. But if you're aiming for distinction, you probably want to give that kind of more complicated reality a go and try to understand and wrap your head around that. So, as I said, we're starting with kind of more simplified version of DNA and RNA. And you will realize that DNA and RNA are both types of molecules called polynucleotides. Now, the word poly in that term refers to many. So poly simply means many. And so it suggests that DNA and RNA contain many nucleotides. And so nucleotides are these smaller repeated molecules. When you put them in a string, that's what RNA and DNA is made out of, essentially. So in case of DNA, you have two strings of nucleotides running side by side, a bit like two trains made out of multiple carriages sitting on parallel tracks. Whereas RNA is a lot shorter, and also it contains a single string of these nucleotides. So don't worry about this diagram too much, but I wanted you to have a kind of quick overview of what we're trying to describe. So we're going to go and describe this diagram in a lot more detail further on, so don't sort of panic about looking at it and think, oh, I don't really understand what it means. Sometimes DNA is described as a ladder. And if you look at a picture of a ladder, you can see it's got two sides, and then you've got these kind of connections in the middle, the rungs of the ladder, which you use to climb up and down. And so hopefully you can see that if you compare this picture of a ladder to this diagram of DNA, you see why there is this reference. So you can see you have two sides made out of these sugars and phosphates, the circles and the pentagons. And then you also have these connections in the middle, which give you that kind of ladder effect, the rungs of a ladder. So what we'll do next is we're going to look at the structure of these repeated molecules. And I hope you can see that there is some pattern, some repetition. We have the same shapes appearing again and again and again. And so these repeated molecules are known as nucleotides, as I mentioned previously. So any nucleotide, either in DNA or in RNA, it's made out of three main components. So the first component, and I'd like you to have a go at drawing this nucleotide structure. The first component is usually represented as a circle. The colors, by the way, don't matter. They can change from one source to another source. But certainly the shapes tend to be fairly consistent. So phosphate usually represented by circle, as I said, and these do not change from one nucleotide to the next. All nucleotides have the same phosphate group. Then you've got a pentagon shape, which represents pentose sugar. The word pentose refers to the fact that it looks like a pentagon, so 5 sides, 5 angles, but also it refers to the fact that it contains 5 carbon atoms within its structure. And then the final component is known as nitrogenous base, and it's usually represented by sort of these oblong shapes, sometimes look like a bit like flags. You can see a couple here, for example. That's quite typical representation of bases. But sometimes they're just represented by this kind of oblong square shape. So these are the three key components of a nucleotide. So next few slides, we're going to look at each component in more detail. By the way, if you're starting to get sort of confused or tired a little bit of the input, good idea maybe to pause, have a little walk around, have a cup of tea, and then maybe come back to it and listen from that point, or go back to the start and re-listen to some of the earlier input. So anyway, off we go looking at each of these components one at a time. So the first component we're going to look at in more detail is this pentagon shape, which is your pentose sugar. So as I mentioned, it's a 5-carbon sugar, and depending whether this sugar is found in DNA or RNA, there's going to be a little bit of a difference. So in DNA, this pentose sugar is known as deoxyribose, hence DNA, deoxyribonucleic acid. So the actual name of the sugar is incorporated into the term DNA. And I hope that you should be able to tell me what the name of a sugar in RNA is. So think back about what the letters RNA stand for, and you should be able to work out that the sugar in RNA is actually called ribose. So both of these sugars are pentose, so both of them contain 5 carbons, but they in terms of structure they are slightly different, hence the 2 different names. So if you look at kind of again going from, you remember my very simplified drawing of a pentagon, a bit like that kid's drawing of a house, but you remember there is complexity behind it. So the complexity behind it is that you can actually draw the structural formulas of both deoxyribose and ribose, and this is what they would look like. So you can see that the main difference is just this one atom of oxygen, which is present in ribose and it's absent in deoxyribose. Everything else is exactly the same. And if we count the number of carbons, let me show you this, for example, deoxyribose in more detail. you can see that you have 5 carbon atoms, so they're highlighted by these numbers in red. So 1, 2, 3, 4, and 5. So it would be a good idea to have a go at copying the structural formula of deoxyribose. I suggest, if you kind of feel a bit confused by the whole thing, start off by drawing this central structural circle, that pentagon shape, and then from there start adding other atoms. So another thing which we usually discuss in class is the bonds between those atoms. So each letter represents a particular kind of atom. So for example, C, it's carbon, and capital O is oxygen, and capital H is hydrogen. I hope you're familiar with those. But also you can see that you have a number of black lines, and black lines represent chemical connections, or we call them in chemistry bonds. So these represent bonds which each of those atoms are connected to other atoms to make the whole molecule of this sugar. So you can have a go at actually working out how many bonds each of these atoms have. So for example, if you just look at individual hydrogen atom, like here, you will notice that each of your hydrogen atoms always have just a single bond connecting it to something else. So let's say this one, just one single black line coming off that H, suggesting that it's one bond between hydrogen and something else. So with oxygen, again, if you sort of have a quick look around this molecule, you'll see you'll always have 2 black lines coming off each of the O's, and that shows you that there are 2 bonds between oxygen and other atoms. And then the final one is your C, which is carbon, and you've got 4 bonds. So that gives a kind of pattern of how these atoms are connected to each other, because you have a fixed number, if you like, of bonds which you're able to make between each of the atoms. So another point which I was going to make is numbering of carbon atoms. So there is a rule about how atoms of carbon are numbered in sugars in organic chemistry. So you always start with going from O in that from that oxygen in the pentagon, and you move clockwise counting your carbon. So that means that is carbon 1, 2, 3, 4, and 5. And these numbers are going to be important for us further down the line when we're looking at the direction of DNA strands, because they're often used in books to describe direction of the strands. Now, if you are finding it hard, don't forget that certain part of this topic are only expecting for distinction grades. So you remember what I said, if you want to keep it simple, just remember that pentagon is representing that pentose sugar, and you don't have to go any further and worry about numbering of carbon atoms, if this is a little bit kind of beyond you, if you like. So moving on to the next component. So the next component is phosphate group, as I said, in most cases represented by this little circle. And the importance of these groups is to link one nucleotide to the next one. So you have these alternating phosphate groups and pentose sugars going on to connect one nucleotide to the next one. So you can see it here how one phosphate hooks onto the next, is hooked onto the sugar in one nucleotide, and then from the sugar you are getting connection to the next phosphate group. So that is sort of becomes the side of your ladder, which is holding the whole of the strand together. And these alternating sugars and phosphates are known as sugar-phosphate backbone. and then you have bases kind of coming off that sugar phosphate backbone. That's what creates your polynucleotide. The name of the bonds between sugar and phosphate is known as phosphodiester bond, which you might want to write down somewhere. You don't have to memorize it, just be aware that that's the name of a bond given, and it's a type of covalent bond. So phosphate group, again, you remember that behind that simplified circle there is actual structural formula. So that will be the structural formula of a phosphate group. Letter P stands for an atom of phosphorus. And again, if I would ask you to count the number of bonds between phosphorus and other atoms around it, I hope you can have a go at counting and come up with the number 5, because we can see that there is one connection there. So that's 1, 2, 3, and then double bond, but you're counting each connection as a separate. It's 1, 2, 3, 4, and 5 bonds. Okay, and then the final component in a given nucleotide is nitrogenous base. So nitrogenous is because it contains an element called nitrogen, and you have 4 variations of bases in DNA, and these are known as thymine, usually abbreviated to capital T, adenine, capital A, cytosine, capital C, and guanine, which is capital G. And in RNA, they're very similar apart from one difference. So instead of thymine, you have a different base called uracil, or again often abbreviated to just a capital U. So these are variations in bases you're allowed to have in DNA and RNA. So this next few slides about bases are again aiming for distinction grades rather than pass, or let's say merit, depending on how well you describe it. So for as far as merit is concerned, you can just keep it at that level and be able to name the bases. But for distinction, I want you to also be able to use these terms, purines and pyrimidines. So there are 2 categories of nitrogenous bases. The first category is called purines, and these include A and G bases, and pyrimidines, which include T, C, and U. And here I've got a slide which shows you structural formulas of these 2 types of bases, purines and pyrimidines. And I hope you can look at this slide, and without kind of getting overwhelmed with how complex things are, but just if you glance on it and think, well, why do you think somebody decided to put adenine and guanine in one category and the other 3 into separate categories? So if you just look at overall look of these molecules, what you might notice, and this is sort of the standout difference, is that in case of pyrimidines, each of them contains this one ring, if you like, in the middle. You can see there's one here, and thymine also has got one structural ring, and the same goes for cytosine. Whereas adenine and guanine actually have got 2 structural rings. So it's not just one circle in the middle like these guys do, but you have 2 circles side by side, or 2 structural rings. So that's really the main difference. So somebody glanced at those molecules and decided, well, let's put them into these 2 separate categories. So purines have got 2 structural rings, and pyrimidines have got a single structural ring within its molecules. So my next question would be, so looking now at this diagram, what term do you think I could use to describe what's on your screen right now? Now, I know you might look at it and feel a little bit overwhelmed. Well, this just looks like a little monster, right? Something really, really complicated. But actually, when we simplify this drawing, it literally represents a molecule, a structure which we named previously multiple times. So it's a one-word answer, and this one-word answer describes what you can see right in front of you. So if you don't mind pausing the video and just having a little think about what would be the answer, which word can I use to describe this diagram? And the answer is simply nucleotide. That's all it is. So if you think about this molecule, which is in yellow box, well, this is my phosphate group. This molecule here, which has got this characteristic pentagon shape, well, that's one of the sugars, right? In this case, it's deoxyribose because it's got only one hydrogen and no oxygen here. And finally, this little molecule inside the blue box, well, that's one of the nitrogenous bases, cytosine. So if you think back to what I was saying, nucleotide contains phosphate group, pentose sugar, and a nitrogenous base, and so therefore I can represent this structure by a simplified diagram which I showed you earlier on this slide. It's just that one. So phosphate, sugar, and base, that's what makes nucleotide. But this particular diagram shows you, that complexity, if you like, behind a given nucleotide. So this is one of the nucleotides, and this is just a different nucleotide, right? So we still have phosphate group, still have got pentose sugar, but instead of cytosine in this case we've got guanine. So that's a nucleotide containing base guanine. If you'd like to have a go at a representation of a nucleotide, you can pause here and just follow this set of instructions. Use your smartphone rather than your laptop to do it, because then you can use your finger to rotate your model of a cytosine nucleotide, which is quite a nice little thing to be able to do. Okay, so going back to the diagram I showed you earlier on, so hopefully this diagram should make a little bit more sense to you. So this is one strand of DNA on this side, and then the second strand on the other side. And you can see that the bases, which stick out a bit like those rungs of a ladder, they are connected together, and they're connected by a particular type of bond called hydrogen bonds. And another really important thing to notice about these diagrams is that there is a particular way in which the bases are paired up. So adenine or A is only allowed, if you like, or able to bond with thymine or T, and cytosine or C is only allowed to pair up with guanine or G. So that is an important rule to remember, and this rule is known as complementary base pairing. So we've got these two polynucleotides which are connected together through bases, and once they connect, then the whole structure twists into this kind of spiral shape called double helix. So this is another representation of DNA. So you can see here the bases being paired up, A to T, C and G, and then the whole thing is twisted up into this structure called double helix. So one of the questions I sometimes ask is, on this diagram, what is being represented by this brown kind of ribbon shape? And the answer is that those ribbons represent your alternating phosphates and sugars. In other words, it's the sugar phosphate backbones, which are shown as brown ribbon. And then the bases are shown as those connecting colored shapes. So yet another diagram showing you flattened out DNA molecule. So again, it shows you those connections between thymine and adenine and cytosine and guanine. And it also gives you extra bit of information about the number of hydrogen bonds between them. So here you can see that between T and A you have 2 hydrogen bonds, two dotted lines, and between C and G you've got 3 hydrogen bonds. Now, hydrogen bonds are not strong, so they're relatively easily broken apart, unlike those covalent phosphodiester bonds between sugar and phosphate, which are really, really strong. So hydrogen bonds are much weaker. Now, another fact which I wanted to draw your attention to at this point is the fact that the two strands of DNA, they are relatively upside down to each other. So if you look at how sugar, for example, is positioned here, so on this side, the pentagon shape, if we imagine that being like a shape of a house, let's say, so the roof is pointing upwards, right? So the pointy bit is going towards the top of the slide. Whereas if I look at the other side, you can see that that pointy end is looking down. So it means they're not mirror image of each other, but rather they're something called anti-parallel. So they run in opposite directions. So going back to my rule, so the complementary base pairing states that adenine always base pairs with thymine, or it will be uracil if you're talking about RNA, and cytosine always base pairs with guanine. Now, why is that? Well, again, this question, why, at the bottom of the slide, this is distinction level information. So if you're finding it hard as it is, you remember you don't have to talk about purines and pyrimidines. But those of you who aim for distinction grades, you might want to consider this question. So in a minute, I'm going to go back to slide 19 of a PowerPoint and look at the structure of purines and pyrimidines. So why do you think A always pairs up with T or U and cytosine always pairs with G? So let's go back then to slide 19 and see what that slide was showing. There it is. So I've got this structures of purines and pyrimidines. So my first question is, let's say I wanted to reword the rule which says A to T, C to G, and incorporate the words purines and pyrimidines into this rule. What should the rule say? What am I allowed to do as far as purines and pyrimidines are concerned? How am I allowed to pair them up? So let's have a think about that first. Well, the answer to that first question is that quite simply, what you are allowed to pair up is that you're allowed to pair up one purine with one pyrimidine, right? So remember the rule, it's A to T, so it'll be that one with that one, or in case of RNA, it'll be adenine with uracil. So it's one of those and one of those, and then guanine pairs up with cytosine. So again, you've got that rule of one purine being able to pair up with one pyrimidine. So in other words, every time you're pairing up base pairs, you're always pairing up one purine with one pyrimidine. Now, the next question is, well, why is that the case? Why couldn't I have a DNA molecule where at one point I'm pairing up two purines together, at another point I'm pairing up two pyrimidines together, and yet at a different point I'm pairing up one purine and one pyrimidine? Why would not this work? And again, I'd like you to kind of pause and have a think about it before you move on and find out what the answer is. Well, the answer is simply the fact that you remember DNA needs to look a little bit like a ladder, right? So we've got two sides of a ladder and then the connections between these two sides. And therefore, I need to have equal spacing between each side of the ladder. I can't have a ladder which is very narrow at one point and much wider at a different point, and again narrow at another point. So it's got to be consistent throughout, right? And if I try to pair up two purines together, it would give me four structural rings between the two sides of this imaginary ladder. Or if I was pairing up one pyrimidine with another one, then that would give me just two rings. So I can't do that. I can't have one place where the spacing between two sides is much wider than at a different place. So you have to be consistent. So it's good to have three rings at any given point between the two sugar phosphates backbones. So it's good to have two rings from a purine and one ring from a pyrimidine at a consistent space. And that's what this slide you will see is saying. So the reason for that is because there is exactly enough room for one purine and one pyrimidine between the two sides of that ladder of DNA, between the two sugar phosphate backbones. And that gives you exactly enough space for three structural rings, and not more, not four, not two, not one, exactly three. And that's what this diagram represents. You can see here you've got your DNA being paired up, giving you three rings in total, and again GNC being paired up, giving you again three rings in total. So you have this consistent spacing. And then TNA has got two hydrogen bonds and CNG have got three hydrogen bonds. So if you are able to describe this diagram and in reasonable amount of detail, this essentially means that you understood the structure of DNA. So again, that's the rule, just to sum it up. And finally, we're going to have a look at a video which gives you ability to visualize structure of DNA in 3D.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of four possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides.
By the way, hydroxyl group, this is not something I mentioned as a term before, but it simply means that OH um, um, bit on ribose, whereas in deoxyribose you only have H and have O missing. That's why she's saying that hydroxyl group is missing.
Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs.
So I'm going to stop this video because that's pretty much covers everything I need us to cover. Just go back to the PowerPoint. So just to point out one last fact which was mentioned in the video is the fact that you use these numberings of carbon atoms, which I mentioned earlier on, to describe the direction in which strands are running. So going back to this slide, slide number 15, let me just enlarge it. You can see that on this diagram you have number 3 and number 5 given to you, and these numbers refer to numbering of carbon atoms. So you can see that this carbon atom number 3 is connected to phosphate group of the next nucleotide, and within that next nucleotide carbon number 5 is linked to the phosphate. So that gives you that kind of numbering shown in red here. So therefore, if I wanted to tell a different scientist, hey, I'm going to look at this DNA strand in the direction from the top of the slide going towards the bottom of the slide, the way I could describe it is by writing that I'm looking at it in the direction of 5 to 3. So 5 and then dash 3. So that means that I'm moving from 5 to 3, 5 to 3, 5 to 3. Now, if I wanted to describe the opposite direction, let's say I'm looking at the same strand, but now I'm considering moving from the bottom of the slide towards the top. In this instance, I would be describing it as going from direction of 3 to 5. So from direction of 3 moving towards 5. And hopefully you can see why. So it's 3 to 5, 3 to 5, 3 to 5, 3 to 5. So it just gives you that kind of ability just to describe the direction of DNA. And this is, as I said, is important to be able to do so because the two strands actually move in opposite directions, like shown here. So my little task for this slide would be for you to figure out if I am moving from the top of a slide towards the bottom of a slide on both this strand on the right and the strand on the left. How would I describe it? So for this strand, would I say that I'm moving from 5 to 3 or is it from 3 to 5? And the same question applies to here. Remember, in both cases, I'm moving from the top of the slide towards the bottom of the slide. So I'd like you to have a think and try to figure it out. So this directionality of DNA strands, you only need to understand that if you're aiming for distinction. If you're finding it, it's already confusing and too hard, then just skip that bit of information. So that's it. That's the end of my video on DNA and RNA structure. So the next topic we're going to look at will be DNA replication. Thank you.